Hello, everyone, and welcome to another discussion that we're going to have in microbiology. Today, what we're going to do is go over the Chapter 6 activities. In Chapter 6, it talked about all the requirements that are needed for microbial growth. It discusses different types of culture media, it discusses different types of preferences for temperature and pH. So, that's kind of in the first part of the chapter. The second part of the chapter talks about how we quantify microbial growth. So, we can use qualitative methods or quantitative methods where we can actually individually count them, or we can use more indirect methods, just kind of looking at turbidity. And then the very, very last part of chapter 6 is talking about how we can calculate microbial growth. So the activities that we're going to go over today will be discussing that entire gamut of topics there. So without further ado, let's go ahead and dive into our chapter 6 activities. So we have this culture media on our concept map here. We're talking about all the different culture media requirements that are needed for microbes to grow. So we have things like selective and differential, such as McConkey's agar. And remember, like what we've talked about in lab class, McConkey's agar, as well as EMB plates and MSA plates, mannitol salt agar plates, they not only are selective, where they encourage the growth of certain organisms, some gram positive, some gram negative, but they are also differential in that they allow for visible differences between microorganisms to be seen. So for selective media, they inhibit unwanted microorganisms and encourage the growth of others. And safford robe auber is an example of that. Differential media is just defined as being able to show visible differences between the microorganisms. An example of differential media would be fermentation broth. So all of those sugar tubes that you've looked at in class, lactose, galactose, mannitol, sucrose, glucose sugar tubes, they all start off that nice red color. And if the microorganism that is inoculated into that tube, if it's able to undergo fermentation and give off these acids as a byproduct of fermentation, then it will turn that nice red colored tube a beautiful yellow color. Um, Differential media can also be blood agar, and remember we talked about the different types of hemolysis or breaking down of red blood cells. It could be alpha, beta, or gamma. Gamma hemolysis means that there was no breakdown of the red blood cells and the microorganisms just kind of grew on the plate. Beta hemolysis means there was a complete breakdown of the red blood cells, and then we had alpha hemolysis, which was just an incomplete breakdown of the red blood cells. Not only is blood agar considered to be differential, it's also a type of enriched media, and it is most useful for the growth of fastidious organisms or microorganisms that normally can be found in blood samples. And then we have general purpose media such as trypticase soy agar or TSA plates, and they're just general. They'll kind of grow anything. There's nothing in it that's going to show visible differences. It's not going to select for some organisms and inhibit the growth of others. It's just kind of just a nice general media in order for us to grow our microorganisms. And then we also have nutrient broth as a great example of that. For my class, almost all of our cultures come in just regular good old fashioned nutrient broth, or they're on some sort of agar plate. All right, so let's continue and move on down here. So for part A in microbiology lab, you make three different culture plates, one general purpose plate, one selective media plate, and one differential media plate. Unfortunately, you forget to label the plates and you don't know which is which. To determine the identity of each plate, you inoculate it with two different strains of laboratory bacteria. Which of the following results would indicate that the plate you used was a general purpose media? So, since it's just general purpose, the organisms that we grow there should both just kind of equally grow. They both should, there shouldn't be any differences in coloration of the media. That shouldn't turn a yellow color or more of a lighter blue color or anything like that. They're going to grow equally well because it's just general purpose media. Next one here. Um, After inoculation, you see that the particular plate has allowed both organisms to grow. However, one has changed color. Which one of these plates do you think it is? So because we can now see differences between those two groups of organisms that are found growing on that plate, it is a very good indication that this is differential media. Part C. Which of the following types of culture media would best be used by a veterinarian to grow any bacteria in a blood sample? Well, since we know the bacteria can normally be found in a blood sample, where would we go to find the media that would support fastidious growing microorganisms? Well, blood agar is a type of enrichment media. Next question here. Which of the following would you expect to be a function of McConkey's auger? So if we just went back to the concept chart that we have here, we see McConkey's auger is both selective and differential. So all of those things that hold true for selective media and differential media would hold true for the McConkey's auger. So let's look at some of the options that we have here, and we're going to select all that apply. So McConkey's auger inhibit the growth of certain bacteria. Well, yes, remember has that selective quality about it. In fact, McConkey's auger is selective for the growth of gram negatives, and it inhibits the growth of gram positives if it's not a heavy inoculum. Allow for the growth of all organisms in the sample regardless of nutritional requirements. Doesn't really work because it's selective and differential. Change the color of some organisms based on their metabolic characteristics. That is that differential aspect or property of McConkey's auger. So that holds true. Allow for the growth of all organisms in the sample that require only the most common nutrients. Well, both B and D kind of sound the same, and that sounds more like just general purpose media, where McConkey's auger is not general purpose. It is selective and differential. All right. So understanding the bacterial growth curve. So bacteria reproduces by a process called binary fission. The length of time it takes for a cell to complete binary fission and form two cells is generation time. Generation time can also be defined as the length of time required for a population to double. So in this generation, this bacterial growth curve, we have this phase at the very beginning where the numbers of cells aren't really rapidly increasing in the population. They're kind of metabolically getting used to the nutrients that are in the environment, and they're slowly getting their numbers together. So we call that the lag phase. But then once the microorganisms start to exponentially increase, where the population size literally doubles every generation because one cell becomes two cells in binary fission, then we go through what's called a log phase, and that's just exponential or rapid growth of the population. So at some point, the numbers of bacteria that are coming into this population can no longer be sustained, especially if it's in a closed area such as a test tube on an agar plate. And the reason for that is that those numbers can't be sustained is that the population numbers get so big, A, they start to run out of resources, and B, the toxic byproducts of their metabolism are kind of polluting the area. So we end up with a situation where the number of microbes that are coming into the generation that are new microbes are roughly equivalent to the number of microbes that are leaving out of the generation. So at that point, we say that the population is stationary. We call that our stationary phase. And then when we have the amount of nutrients in this closed system are no longer available, they start to die off, and we have just this accumulation of byproducts from fermentation, and they're pretty toxic to those new cells coming in. If there's no way to put new food into the system and get rid of all the toxic waste, then what we see at the bacteria that will go through what is considered a death phase, or that's the exponential decrease in the numbers in the population. So when we look at this and we try to put these labels on the chart, what we could do here is that, and I'm going to go ahead and go to slightly different, and go look at this right here. So what we see here is that we have the log number of bacteria over here on the y-axis, and then on the x-axis we have time. So this is happening over time. So what's happening here is that at this lag phase, the bacteria are metabolically active, but they're not reproducing as quickly, so their numbers are not increasing super fast, because this is where we're going from one cell to two, and two to four, and four to eight, and those numbers just aren't getting super huge yet. But at some point, we have this rapid exponential growth that we call the log phase. And then here is the stationary phase, where the population growth has halted. The number of new cells coming in roughly equals the number of cells that are dying. And then we have this death phase where it's the exponential decrease or decline in microbial cells. And remember, this holds true in a closed system where there's no new nutrients coming in and there's no way to get rid of waste. That curve can change and can be manipulated if we are inputting new nutrients or if we're getting rid of that waste. So, part B, when bacteria are inoculated into a new sterile nutrient broth, their numbers don't begin to increase immediately. Instead, there is a lag phase that may last an hour or even several days. Why don't bacterial numbers increase immediately? Well, the reason they don't increase immediately because they have to kind of get used to this new media. So, if there are specific types of enzymes that need to be manufactured or different amino acids or growth factors, all of those have to be made first in order for the cells to utilize the nutrients that are in that media. So the best answer we have here would be letter B. The microorganisms have to adjust to the content in the new media, making necessary amino acids, growth factors, or enzymes. So A here says the bacteria have to establish a biofilm, and no, they don't have to attach to anything in order for the numbers to increase. Letter C says there aren't enough nutrients for the bacteria to grow, and the growth is delayed until there are some dead cells to cannibalize. Well, they don't cannibalize cells. They actually just use the nutrients in the media, so that one doesn't work. D, the medium contains inhibitors that prohibit rapid growth of the bacteria, and these must be inactivated before bacterial numbers will increase. Well, if there are inhibitors in the growth media, then the microorganisms just won't grow at all until you remove that. So that doesn't work either. So our best answer is B. Going on to part C, after a period of rapid growth, bacterial rates slow down. They get into the stationary phase, and that's where the number of cells coming in roughly equals the number of cells that are dying out of the population. Select each statement that accurately describes what's happening to bacterial cultures during the stationary phase. So, what we have here, first of all, are the number of cells that are dying balanced by the number of cells that are being formed. Absolutely, that is something we talked about. Harmful waste products may be accumulating. Absolutely. We have this large amount of waste products because we're in a closed system and there's no place for them to go. Cells are not increasing in numbers because they have not yet adjusted to the nutrient availability. Well, at this point, they've not only gone, this is something that happens in the lag phase. We've already gone through the logarithmic expansion. So we've gone through the log phase and now we're at stationary. So that one doesn't really work for us because we've already adjusted to the nutrients available. The cells are dead, therefore the numbers are staying constant. No, not all of them are dead. We're still reproducing, but the number of cells coming into the population roughly equals the number of cells that are leaving. And then the final one here, the cells are likely running out of nutrients. That is something that we have very much discussed as a possibility or what's happening in this closed system and why our numbers are not just rapidly, steadily increasing, that we see that they're kind of leveling out. So, prolonging exponential growth, how can we do that? So, a chemostat is a continuous culture system that is designed to promote and prolong exponential growth and prevent bacteria from entering a stationary phase. What would need to be the mechanisms through which this could work? So, we've already discussed that bacteria will go into this kind of stationary phase, and I'm just going to slide up here a little bit so we can see that because harmful waste products may be accumulating that we discussed here, and the cells are likely running out of nutrients. So if those two things could be what puts a bacteria population into stationary phase, how about if we remove those two obstacles? You're absolutely right. That would absolutely allow these microorganisms to stay in this logarithmic exponential growth. So what this chemostat should do, it should do two things. It should get rid of waste products and it should provide a continuous food source. So let's see which of our options give us that. A, chemostats promote the formation of endospores, which enable long-term survival of bacteria. Although endospores do enable long-term survival of the bacteria, not all cells, not all bacteria are capable of forming endospores. So that one doesn't work for us. Chemostats include growth-promoting agents that prolong microbial growth. Well, that would be good, but that's really just answering one portion of the problem. We don't know how we're going to get rid of all these waste products that are accumulating in there. So although B sounds nice, it's not really addressing both sides of the issue that we're having. So letter C, chemostats provide a continuous source of fresh nutrients. Okay, We like that. And remove waste and dead bacterial cells. Bingo. Those are two of the things that we said need to be addressed in order for chemostats to effectively keep this microorganism's population in a logarithmic expansion or continuous growth. And then let's just look at D to kind of see what that says. Chemostats include detoxifying agents that inactivate growth inhibiting toxins that develop during continued microbial growth. Well, that's just addressing one aspect of it, the waste. We still need continuous fresh nutrients coming in. So now on to part E, where part E we're going to start talking about, and this is a part in your textbook where we start to talk more about calculating microbial numbers or calculating generations. So we're going to go back over here to this, and we're going to start talking about that a little bit. So we've already looked at this, those different phases that we've seen. And this animation I will link in the description that you're able to watch this animation if you so choose to. And it's just kind of showing that one cell becomes two. So it's kind of that logarithmic expansion. And in fact, let's go have a look-see at it. It's pretty short.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly.
All right. So that was a nice little short video. So, with binary fission, one cell always becomes two. Never becomes more, never becomes less. One cell makes a copy of itself and it becomes two. So, when we just have one cell and it has not made a doubling of itself, it is just one cell. What this chart is showing us is it's showing us population growth of bacteria in a couple of different ways. Now, if we have two cells, this two is going to remain constant because one cell becomes two. So we have the number of cells there are two, and we did the visual representation of that. We have two on there. So the numbers that are the exponents here are representing the number of generations. So in generation 0, there was just that one cell. In that first generation, that one cell became two. So that's the first generation. This is like the grandfather, if you will, or the great-great-great-grandfather when we start. So this one cell, if it makes a copy of itself in the next generation, it becomes two cells. This first generation, so 2 to the first power is just 2. If those two cells, go ahead, so these two cells here, if they go through binary fission, one cell will become how many? 2. It'll become these two. This one cell will become these two. So when we get to the second generation, we'll have 4 cells. And an easy way to calculate this, because right now the numbers are pretty easy, right? It's like, oh, okay, one cell becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 64. Like those are reasonable numbers. But what happens when we get to the 10th generation? Can you see how easily those numbers are going to get like way out of control? So an easy way to handle this, and notice that this base number always stays the same, that when you're trying to calculate the number of cells in a generation, that we can just use 2n, where n equals whatever number generation we're in, whether it's the first generation or the 15th generation. So this is just a real quick, dirty, and easy way, and I apologize for my handwriting with a mouse. It ain't pretty. So what we can use then is that we can take this 2n and we can plug any number into this. And we could figure out how many cells are in that generation. So if we have the 100th generation, well, we just put 2 raised to the power of 100th on here. So that's a very easy way to kind of use that. So there are a couple of different formulas that I want you to remember, and this is kind of the first one that I want you to remember, 2n. All right. So now let's go on to our, oops, went too fast. So now over here, what we have is, so you kind of recognize this chart. This chart really isn't any different. It's just this time it tells us the number of generation that we're in, and then it will tell us the number of cells that we have. And then we have a little bit of a new sort of column over here. This new column is just going to make it easier for us to chart those number of cells. So same scenario, at the 0th generation, you just had 1 cell there. When you get to the 5th generation, there's still that 2n. So if you have your calculators available, if you take 2 to the power of 5, that gives you 32. 2 to the power of 10 gives you 1024. And you can actually, if you want to pause the video and look at how that process works, you'll very easily see like, oh, okay, this all makes sense. Now notice when we get to the 20th generation here, we're looking at 1 million cells. Can you imagine trying to plot 1 million on a graph? That's going to be nuts. So in order to help us better get a handle on these cells and how to quantify them and how to represent them on a chart, we're just going to use the log of that. We'll just use the log base 10 of that. So if we took like the number, we'll just use the number 32, and we did the log of 32, we'd see that it's 1.5. It's actually 1.505, but 1.51. It's just 1.51. So if we did the log of this number here, so if we did the log of 524,288, the log of that, 5.7. So very easy to do this. If you have an iPhone, and I only know iPhones, I don't know Androids, I apologize. If you have an iPhone and you kind of turn it to the side in landscape mode, you'll get a scientific calculator. So when you open up your calculator and you turn your iPhone to the side, and I'm going to just kind of show you guys what I'm talking about here. So this is my iPhone, and when I turn it up this way, this is just the normal portrait and just gives you kind of just a generic little calculator. But if I turn it in landscape mode, it gives me a scientific calculator. So that any number that I want to use, so let's say that we'll put 2 in here. If I go over to where it says log base 10 and I hit that, it will give me the log of that. So I kind of hit too many things. So let's try that again. So if I hit 2 and then I hit log 10, and that gives me the log of 2. So any number that I want, I'll just punch that number in. So let's just punch in 42, and then if I want to hit log 10, it'll give me the log of that number. So that's how it works for iPhones. I am really not sure how it works on Androids, but I'm sure it's pretty similar to that. If you have a scientific calculator, I'm sure you have that log function as well on that. All right, so the log of the numbers over here, it equals the same thing. They all equal the same thing. It's just that these are a much easier way to kind of calculate this. So remember, the first formula that I wanted you to know was 2n, where n equals what? The number of generations. So we can plug any generation number in there, and we're going to get how many cells are in that generation. Okay? So here's just a really nice picture of showing how when we use the actual number of the cells, what that graph looks like, as opposed to using the log of the cells. It's just a nice cleaner graph. It's much easier for us to get a handle on. So we've had our first formula here. Check 2n, where n equals the number of cells present. or 2n equals the number of cells present in that generation, where n equals the number of generations. And another way that we can say generations, and you will see this in the activity that we're getting ready to go over, is that we can say doublings, we can see replications, we can say divisions, we can say generations. So anytime you see those terms in the question, you should be thinking, well, I could probably use 2n, that formula, to figure out exactly how many cells are in that generation. Or after 5 doublings, a question might say a cell has undergone 5 doublings. How many cells are in the population at that point? Well, you can use 2n because the 5 doublings are what you would put as that exponent. That's what you would put in the place of n. So let's work on a question to look at the other formulas that I want you to know. So if you have 100 cells growing for 5 hours and they produce 1,720,320 cells, what would be the number of generations and what is the generation time? So the next formula that I want you to have memorized on here is this formula to figure out the number of generations. So this formula here, where it's going to be the log number of cells at the end minus the log number of cells at the beginning divided by 0.301. So you remember in math that whatever units that you have here on the top in the numerator, those units need to also match in the denominator. So we're using the log of the cells at the end, and in this case, that log of the cells at the end was 1,720,320 cells. And the number of cells at the beginning, we started off with 100 cells here. We're going to turn those numbers into the log form. And because one cell always becomes how many? 2 cells. Then we're going to use the log of 2. Take out your calculators and tell me what the log is of 2. And the log of 2 is 0.301. So that's what this is. So this just equals the log of 2. So if you can't remember 0.301 when you're memorizing, because you are going to memorize this formula, when you're memorizing this formula, if you can't remember that log 3.301, then just remember log 2. Because one cell becomes how many? 2. And we want the log of that because we want all of our units to stay the same. So let's go ahead and calculate this first one out. So we had the number of cells in the end was 1,720,320. And if we took the log of that, what you should have gotten was something like 6.23. And then we subtract that from the log of cells at the beginning, and that was the 100. So the log of 100 is 2. We divide that by 0.301. And what's another way we could have written just 0.301? Log of 2. So when we do that math out there, 6.23 minus 2 divided by 0.301 gives us 14.05. So you can't really have like 0.5 of a generation or half of generation or a fraction of a generation. Either it is or it isn't. So we're just going to round this here, that 14.05, and say 14 generations. So that would be the answer to how many generations are in this population. Now the next formula I want you to remember is how to calculate generation time. Generation time is defined as the length of time it took for a bacteria to go through its doubling. So the length of time it took for this one cell to become 2. Some bacteria generation time is as quick as 20 minutes, things like Staph aureus. A lot of pathogens have a very quick generation time. Some things are a little bit slower, like tuberculosis can have a generation time of up to 48 hours or something crazy like that. So generation time differs. But we can take our understanding that one cell becomes 2 and we can figure out how long the generation time is. So for this question here, the formula is we're going to take the time divided by the number of generations. So because most bacteria, especially for what we're going to work with, they have a generation time in minutes. That's why we're going to convert hours to minutes. So this could just as easily just said time. This stuff at the numerator, but we're going to calculate or convert it to minutes simply because most microbes um, generate in minutes. They don't generate in hours. So we have 60 minutes up here. And then the way I like to write these things out for my own, the way that I learned math, and then we're going to divide that by the number of generations. So in this question, we'd already figured out that we had 14 generations. So I'll put 14 down here and a G to keep up with my units. And so the time I have here is that we had 5 hours, and I just got that from the question. So those 5 hours, what we want to do with that is that we want to make sure that our units cancel out. So we have 60 minutes would be in 1 hour, so I'll put 1 hour down there, and I put 5 hours up here. So we want to just be in minutes per generation, so I'll cross out that hour, cross out that hour. So really what we have is 60 times 5 divided by 40. And if we calculate that out, divided by, not 40, 14, we have 21.42 minutes. And we have minutes per what? Making sure that our units are right, minutes per generation. And that would be our correct answer. So when you know these two formulas, and actually 3 formulas, because here's our first one, 2n, that's going to help you to answer any questions that I give you that ask about either calculating the number of cells in a generation, that's at 2n, or calculating the number of generations, that's this puppy right here, or calculating the generation time. You can do that. And for this exam, I will allow you guys to have calculators. I do not expect you guys to do this off the top of your head. If you don't have a scientific calculator and you just have like your iPhone, I will be letting you guys have limited use of your iPhones, but it'll have to be in airplane mode, so I know that you're not going online to look up answers to this sort of stuff. All right, so now we're going to go and answer some questions. We're going to keep those for now. We're going to go back to our activity and answer this question. So, a broth media has been inoculated and microbial numbers will be counted periodically to generate a bacterial growth curve. At 2 hours after inoculation, the culture has progressed through lag phase and is now in log phase. At this point, the population size is 1 million cells. The generation time is 30 minutes. Assuming the continuation of log growth, how many cells would be there at 2 hours of growth in log phase? So the first thing we want to do is ask ourselves, can we use the easier of those 3 equations? And the easiest of those 3 equations was the 2n. So can we use 2n? And I don't know if I can turn this into an exponent. Very nice. It's going to take a while. Can we use that? That's the question that we have. So in order to use 2n, we already have the 2. We just have to be able to figure out what the n is. So, and the n represent what? The number of generations. So we can figure out, if we can figure out the number of generations, then we can just use this very easy formula. And I want you to kind of go through these same steps when you're answering these types of questions. So figuring out the number of generations means that we need to be able to calculate the number of generations you'd get in a set amount of time very quickly and easily in our head. So it tells us that the generation time is 30 minutes. Okay, so it's just a half an hour. And it wants to know how many cells will be there in 2 hours. So 32 hours is a number that, we can very easily figure out how many 30-minute segments are in that. We can do that quick and easy in our head. We know that there are 4 30-minute segments in 2 hours, right? So you have an hour is 60 minutes, and there are 2 30-minute halves in 1 hour. So there are going to be another 2 30-minute halves in 2 hours. So that means that there are going to be 4 opportunities for doublings or generations to be made. How do I know this? Well, generation time is only 30 minutes, and we have 2 hours to get that we want to know how many cells are there in 2 hours. That's in a set amount of time. So we can say that this n is going to be 4 because we have four 30-minute segments in that 2 hours time. So since we can figure out what the generations, how many generations we'll have, because we knew what the generation time was, we knew how many hours we're allowing them to grow, that in 2 hours we can get 4 generations because they're only 30 minutes long. So because we can do that, then we know that this is our n, our number of generations. So when we take and go back to our handy calculator, and remember you can put this in landscape mode to do this, what we find is that 2 raised to the fourth power is 16. Now we're not talking just 16 cells because we started off, remember, with a million cells. So we have to take 16 times 1 million, which is equal to 16 million. And do we have any of that as our option? Absolutely we do. 16 million cells. All right. So going down here to the next one, it says that this question asks you to make comparisons regarding the slope of exponential growth based on generation time. And we are going to do a little bit more math, so don't go too far away. The generation time, so each of the 3 graphs shows below data collected during the exponential growth of a species of bacteria in 3 different growth conditions. Which growth condition represented the longest generation time? So we didn't want to see the highest number of cells. We want to see the longest amount of time. So what A is showing is that it's actually showing just kind of like an increase in the cells at some period of time. So let's look at some other ones. B here, let's pull it down so you can see it better. This, we have a lot of cells happening, but it's in a relatively short amount of time. So it's not that the time was increased, it was just that the number of cells increased. And then when we look here, we have option D, and this is where the time is increasing. The number of cells isn't actually increasing, it's the number of time, and that's what we wanted. So letter C, or letter D, is our correct answer there, that this one is going to be the condition that results in the longest generation time. All right, so counting bacterial numbers, which of the following methods would most be appropriate for getting data to plot bacterial growth? Well, indirect estimates based on turbidity, we're just looking at kind of the quality of the broth and how cloudy it is. It doesn't really give us a good indication or enough information to put on a graph. We need some actual hard data or numbers. Direct microscopic count of it. Remember, underneath a microscope, you're only looking at one kind of quadrant on there. You're not really, and it's just a small sample, so it doesn't necessarily give you a good representation of all the cells that are in the population. Electronic plate cell counter, and once again, it's like a direct microscopic count that is just giving you a representative sample. The best way to do that would be to count the number of colonies on a plate count. Plate count, there we go. All right, and so this labeling of this chart here, this would be your lag phase, this would be your log phase, stationary, and your death phase. And then down here you can write down what each of those represents. That's either from your textbook or from our lectures or even just from our discussion earlier in this video. What results when a single bacterium reproduces? Well, we have 2 genetically identical daughter cells that happen here. Now, they're not always going to stay identical, and we'll talk about in Chapter 7 some of the things that happen to allow for differences to take place. That's what those sex pili are for. We have conjugation, transformation, transduction, all sorts of things that kind of change the genomic characteristics of a bacteria so that they're not all just clones of each other. But when that one cell becomes 2 via binary fission, they're technically 2 genetically identical daughter cells. Which of the following is not a step in bacterial cell division? Well, since they don't have a nucleus, there is no disappearance of the nuclear envelope. And then which step of binary fission is a reason for genetically identical daughter cells? And the reason for that is because we have the replication of the plasmid or the bacterial chromosome that goes from that duplicates itself within the parent cell, and then when that new daughter cell is made, it gets an exact copy of that. All right, so now we're going to have some more questions that we're going to work with here. So what I would like for you to do is that I want you to pause this video, and then we're going to come back and work the solutions together. And then let's see where you went wrong, or what ways that this actually, what kind of answers you got. And hopefully you'll have a better understanding of how to do these. So for my students, you will see some of this in your first short answer question. So you definitely need to know this. So working with number 1 here. So starting with 3 cells, how many cells would result from 3 rounds of replication? So we know the number of generations here is going to be 3, simply because it says 3 rounds of replication. So replications, doublings, generations, those all say the same thing. So we can just use 2n. Let's just write it over here. 2 raised to the nth. So in this case, it's going to be 3 raised to the 3rd here, because we're talking about 2. We're talking about, so that one cell becomes 2, it's raised to the 3rd, and that number is going to give us 8. However, we cannot stop there, because we started with 3 cells. So we now have to take 8 times 3 is equal to 24. So our correct answer for number 1, and we'll make these font colors match, is 24. All right, moving on to number 2. If you begin with 6 cells, how many cells would you have after 3 rounds of division? Well, we once again see that we can say how many divisions or rounds of replication we're using. So 2n is going to work for us. So we'll use 2n. And in this case, 2 is always going to stay 2, because one cell becomes 2. We have 3 rounds of replication that we're working with here. And so this is still going to equal 8. However, we're going to multiply it by 6 cells this time, because that's what the question tells us that we're using. And the answer that we're going to get for that is now we have 48 cells. And I should have labeled this as cells too. And let's make this, and that's not really a bright color, is it? So 48 cells. And then moving on to number 3. 3 cells with a generation time of 60 minutes are inoculated into a culture media. How many cells are there after 5 hours? Can we use 2n? We sure can. We can use 2n because we can very easily and very quickly figure out the number of cells that are in this generation, because they have a generation time of 60 minutes. So very straightforward. After 5 hours, we'll get how many generations? 5. After 6 hours, we'll get 6 generations. So we can still use our wonderful 2n, which makes everyone happy. So the number of generations that we have are going to be 5, because they have a generation time of 60 minutes. So if it takes 1 hour for one generation, then for 5 hours, or for 5 generations, it's going to take 5 hours. So we're going to raise that to the 5th. And what we end up with here is 32. How many cells did we start off with? Well, the question tells us that we started off with 3 cells. So we're then going to multiply that by 3. And our number that we get here is 96 cells. And let's make you purple. Purple works. All right, moving on to number 4. Number 4 says an experiment began with 4 cells and ended up with 128 cells. How many generations did the cells go through? So we want to figure out the number of generations that we have here. So We can't use 2n on there because they want to know how many generations, so we don't know how many generations there are, so we don't know what n is. We have to figure that out. But we do have a number of cells at the beginning and a number of cells at the end. So the only formula that allowed us to use that information was, let's find that puppy right here, was this jobby over here. Number of generations gave log of cells to the end minus log of cells beginning divided by log of 2, or 0.301. So when we come over here, what we're going to do is log of cells at the end, and that was 128 cells minus the log of cells at the beginning, which was 4 cells, and then we're going to divide that by log of 2. So when we do all of that math out, let's get out our handy dandy calculator here and make sure that she is in landscape mode. We had 128, and the log of that was 2.10 minus the number of cells at the end was, or the beginning was 4, and the log of 4, let me make sure I wrote that right, is 0.6020, and then we're going to divide that by log of 2, which was at 0.301. And then when we just do all of that math out, 2.10 minus 0.6020 divided by 0.301, and what we are left with is something that looks like 4.97. And so it's not a 4.97, and what we can do with that is that we'll just round that up to 5. And we have 5 what? Generations. And let's make you a pretty orange color. So it's kind of obnoxious. I apologize. All right, going on to number 5. So number 5 says a microbe produced by binary fission every 20 minutes under favorable conditions. If you started with 1 cell, how long would 10 generations take, and how many bacteria would be present if all conditions were just right? So we could use 2n. And the reason that we could use 2n is what? Well, because it tells us on here, didn't want that color, because it tells us on here how many generations we would get. So it says we start with 1 cell, how long would 10 generations take, and how many cells would be present. So what we want now is that we're going to raise this to the 10th power, and what that gives us is 1024 cells. So we know how many cells are present. Now the next thing that we want to do is that we want to figure out exactly how long it took us to get to 10 generations. So we know what the generation time is. The generation time is 20 minutes. And there are a couple of ways that you can figure this out. Well, 20 minutes is one of those nice round numbers that you can figure out exactly how many 20-minute segments there are in a particular amount of time there. So we know it reproduces every 20 minutes. We want to figure out how many generations are on there. You could very easily just go ahead and write down that, okay, for the first generation that took 20 minutes, second generation that took 40 minutes, third generation that took another, that was at 60 minutes. You can kind of go through all the way and do that. Or what you could do is that you could take that formula that we have, that generation time, and I'm just going to write it down here because I think there's a little bit more room. Generation time equals time divided by the number of generations. So we could use that and we could just kind of move it around a little bit to figure this out. We can just kind of move things around. So we know what generation time is. Generation time is 20 minutes. We're trying to figure out this time here. So we could just select or rearrange this equation to just solve for time itself. So what we could, that'd be a really easy way that we could do it. And what we would get is that you would take this 20 minutes of generation time and then we'll multiply it by the 10 generations that the question told us we already have. And what you're going to end up with is 200 minutes. And we can take that 200 minutes and then we could convert that into hours and minutes, and that becomes about 3 hours and 20 minutes. So we'll just make this color correspond to, we haven't done red. So we'll do red, and we'll do red for this. And we'll do red for that. So what we did on 5 is that we took the third formula that we learned, the generation time, and we just solved for time this time instead of solving for generation time because they gave us generation time. And to solve for time, in order to isolate time by itself, we just, since we're dividing on this side of the equation, we're going to multiply on the other side of the equation. And we took the generation time, the 20 minutes that they gave us, we multiplied it by the number of generations we had, which they told us was 10 generations. And when we just did that math out, that's what we ended up with. So you had 200 minutes was the generation time. And then if you convert that, you can convert that to hours and minutes by dividing it by 60. And then you got 3.33. A third of an hour is like, you can use just 20 minutes, or about 20 minutes, or 3 hours if you want to just keep it there. All right, so for the next question here, this is that serial dilution question. And we've done this before in our class once already. We had a lab on it. If you have not done the serial dilution lab, then you probably are going to start talking about it pretty soon. And with the serial dilution lab, all we did here is that we had our first tube, and we put in there 1 ml of the microbe. So we had a 1 to 10 dilution. And then we put 1 ml of that into another. So that was 1/100th, then 1,000th, and 1/10,000th, and then 1 millionth. And then what you could do is that you count the number of colonies that you see and you multiply it by whatever sample you took it from. So if we took it from this sample here, let's say we have 3 colonies, then we take 3 and we multiply it by whatever that dilution factor was. So if we were in this, what would this be? 1, 2, 3, 4, 5th one on here, we would multiply these 1, 2, 3 colonies by 10,000. And that's how we do that serial dilution for that. And I'm not sure why nothing showed up for this. Yeah, don't know what happened there. But these are the dilution factors for it that you can see over here. So 1/10th, 1/100th, 1 of 1,000, 1 out of 10,000. So what would have to happen is that when we were ready to, I'm just going to get rid of that for a second. For example, if 54 colonies are on the plate of 1 to 1,000 dilution, then we'd count the number 54 and multiply it by 1,000, and that gives us about 54,000 bacteria per mil in that sample. So when we go back to our question over here, A milk sample is tested for bacterial contents in a plate count assay. 1 millimeter sample of the milk is diluted to 1/10 dilution series, like we saw just on the PowerPoint slide. 1 millimeter of the third dilution tube is plated in a pour plate after incubation. The plate has 54 colonies, indicating that the original milk sample a sample contained how many colonies. And when it says show your work, I would want you to draw out those test tubes. And when we got to that sixth tube, not the sixth tube, we got to the third tube on there. So the first tube is, and let's just kind of work that down here. So the first tube is 1 out of 10. Second tube is going to be 1 out of 100. And the third tube is going to be 1 out of 1,000. So we wanted you to look in that third dilution tube. And so we take that 54 and we multiply it. 54 comes from the number of colonies that we see on the plate. And then we multiply it by that 1,000. And that would give us 54,000. So serial dilution problems are actually really quite easy. Of all the math that we've done today, they are definitely the easiest of those math problems. And we'll just turn that yellow. All right. So that's all I have for this. It's pretty long. You might want to review this, especially the math stuff together. I also invite you to do some more of these problems on your own, just to kind of get more practice with it. All right, that's all I have for now. I will see you soon.